Welcome to our review on drug development. When we're actually thinking about where all these drugs that we now use in our modern lives have come from, then the answer is that the vast majority of them actually came from plants in the first instance. So if you think back in time to before we had all of these pharmaceutical companies making drugs for us, then lots of people would use different kind of plants and herbal remedies to treat things. And that's pretty much the origins of where our drugs came from, and that's still true today. So to give you a couple of common examples here, aspirin, that was actually developed from a chemical that we found in bark from willow trees. And digitalis, which we use to treat heart problems, that one came from a chemical we found in the plant foxgloves, which you can see on the right there. Not all drugs come from plants though. Some other drugs we developed from microorganisms. And one of the really famous stories here is the origins of penicillin. What we actually had was Alexander Fleming, who you can see on the right there. Then in his lab, he wasn't the neatest scientist, let's say, and he had a whole load of petri dishes stacked up in a sink, which had been sitting there for a while because he hadn't really fancied cleaning them out. But when he actually came to then clean these dishes out, what he found was that there were some that had this mold growing on them. And when he looked, there weren't any bacteria present. So from that, he then found the penicillin. And that's what we then went on to use as our first antibiotic. These days, we've got these big pharmaceutical companies that are involved in making the drugs on a very large scale to meet the needs of our population. However, the origin of many of these drugs is still a chemical that they extract from a plant in the first instance. When we're actually thinking about developing these new drugs, it's not a case of a scientist finds a chemical they think is going to work and then it just gets sold to the public the next week. There's a whole range of different tests that they have to go through before they're allowed anywhere near the general population. We've got to check them for safety and for their effectiveness. And we can split this up into pretty much three main stages. The first one are the preclinical trials where we're using cells and tissues. The second are the preclinical trials where we're using live animals. And then the third one are the clinical trials on humans themselves. If we consider the preclinical trials on cells and tissues first of all, then within a lab, what we can do is actually have these sections of cells or tissues growing in a petri dish, for example. And what we can do is expose those to our drug that we wish to test and have a look to see what happens. If the drug kills or damages the cells or tissues, then it's not going any further. If, however, there's no signs of it killing or damaging those cells, then we get to move on to the next step. Stage 2 then is where we're going to use live animals to test the drug. And the whole idea behind doing this is threefold. Firstly, we need to test the efficacy. So that just means we're checking to see if it works and has the effect we actually expected it to. Second, we need to know about the toxicity. So we're looking for how harmful it is. When we administer the drug to the animal, does it stop their heart? In that case, we stop the test. And finally, the dosage, so we can actually get the concentration and how often someone takes the medication right before we go on to the humans. It has to pass both of the lab sort of cells and tissues and the animal tests before it gets anywhere near a human. When we're talking about these animal tests, then in the UK, our law actually states that they have to be tested on 2 different live mammals before they can actually be licensed. And there are obviously some people who disagree strongly with animal testing because they say it's cruel. There are other people who say that testing on animals is actually a pointless exercise because they're so different to humans. What are we really learning here? The argument that goes for animal testing, though, is we need to know what these drugs are going to do to a living organism that's actually fully functioning in the first instance. And we can't really just roll out these tests on humans without obviously knowing that in the first instance. Assuming our drug has got through those preclinical trials, then the next step are the clinical trials. Now these are again split up into mini stages, and the first one of these is to test it on healthy volunteers. And the reason that we're using healthy people with no disease that we might be looking at is for the simple fact that we want to know, are there harmful side effects? So what we do is we give our healthy volunteers a low dose initially and check to make sure they don't keel over, and then after that we will increase the dose gradually to see what happens. If there are quite serious side effects, then obviously we'll stop that trial. If there are only minor side effects, then we just write them down and make a nice list of the potential side effects that we could get. Once we've checked our healthy volunteers with the drug, the next step is to test it on people who are actually suffering with the illness that we're trying to find a drug to treat. And the whole idea behind this is to allow us to find the optimum dose. So what this is the dose that offers the best effectiveness, but it limits the side effects. Because it might be fabulous that if you take a really big dose of this drug, then it's going to eliminate the disease incredibly quickly, but you may also end up with debilitating side effects. That's not how we do things. So it's getting the balance between the effectiveness and limiting the side effects in the person. When it comes to using humans for these trials, then we split patients into two groups. One of the groups will receive the drug we're testing, and the other will receive something called a placebo. Now, a placebo is a replica of our test drug, but it just has no active ingredients. So it's kind of like a sugar pill, really. The whole idea behind doing this is to allow for the placebo effect to be accounted for in the results. Because what happens is when you give people medication, then sometimes they just think they're going to get better, so they show improvements, even though you might not actually have anything that's doing something for their particular condition there. So by giving one the placebo and one the test drug, then they can all sit and think they're getting a medication that's helping them, and it eliminates that problem where if we only had one group getting the test drug and the other just sitting there knowing they're not getting anything, they clearly wouldn't be feeling any better. So that just eliminates the placebo effect. The other aspect we need to consider when we're thinking about drug trials is what bias there could be. So the way we try to restrict this bias is by using either blind trials or double-blind trials. A blind trial is where the patient doesn't know if they're getting the placebo or the drug, but the doctor does. And a double-blind trial is where neither the doctor nor the patient know who's getting the placebo and who's getting the drug. And the only time they find that out is after the results have all been submitted. And as I said, the whole idea behind that is to reduce bias in the results there. Finally, when we've gone through all of these stages of drug testing and you think you've got something that works, then you have to submit your results for what's called a peer review. And the whole idea of peer review is to try to prevent scientists making false claims. So when you submit your work for peer review, other scientists are going to check that your work has been carried out rigorously and that it is valid. So they don't just let anything go through, because otherwise people could just sit there and claim they had a new wonder drug that does all these amazing things. And if it wasn't checked, it could do absolutely nothing. So peer review is just used to prevent those false claims. Hopefully at the end of this video you can describe how we develop new drugs. So you can explain what happens in the different steps of testing the drug, either in the preclinical trials or the clinical trials. You can talk about the placebo and placebo effect, and also the importance of peer review.